Welcome to Biology at Priyajit, my dear viewers. Today we will discuss one of the most important classes of biomolecules, the lipids. Just as a building needs walls, insulation and energy supply, a cell depends on lipids for membrane structure, energy storage and vital signaling functions. So, from simple fatty acids to complex phospholipids and regulatory steroids, these molecules play a central role in maintaining life at the cellular level. Let, let's understand their structure and biological significance in a simple and symmetric way. Introduction. What are lipids? Lipids are basically a chemically diverse group of biomolecules found in all living organisms. The most important common property of lipids is that they are insoluble in water, which makes them hydrophobic in nature. So, however, lipids are soluble in organic solvents such as ether, chloroform, benzene, and acetone. Major functions of lipids. Lipids perform a wide variety of essential biological functions like energy storage. We know that fats and oils act as the main long-term energy reserves in our body. Structural role. Phospholipids and steroids are major components of biological membrane. This is the biological membrane structure. And thirdly, regulatory and functional roles like lipids acts as enzyme cofactors, electron carriers, light absorbing pigments, protein anchors in membranes, chaperones for membrane protein folding, emulsifying agents like bile salts, hormones and other intracellular messengers like neurons, etc. Fatty acid basic structure. We know that lipid basically made up of fatty acid and glycerol. So the fatty acid is an important component of the lipid, which are carboxylic acids with a hydrocarbon chain and the chain length from C4 to C36, and there should be a carboxyl group, that is bond COOH at one end. So they are the building blocks of fats and oils. Types of fatty acids. Saturated fatty acids, there is no double bonds, so hydrocarbon chain is straight and flexible and it packed tightly, solid or waxy at room temperature. Unsaturated fatty acids, one or more double bonds should be there, and mostly it is cis configuration in nature. Cis double bond creates a kink in the chain. It pack loosely and it make liquid at room temperature. Cis versus trans fatty acids. Cis fatty acids naturally occurring, they are healthier for the body. Trans fatty acids, they are produced by fermentation in ruminants like milk, meat, etc. Industrial hydrogenation of oils found in French fries, donuts, cookies, etc. We know that trans fatty acids are more harmful than cis fatty acids, because in trans fatty acids there are hydrogen bonds, hydrogen atoms are present at the opposite side, but in case of cis fatty acids they are present on the same side. So the trans fatty acid can create a kink, but cis fatty acid in general, they do not create kink or bend. So we know that industrial hydrogenation of oils, they produce trans fatty acids. They are found in French fry, donuts, cookies, etc. The health effect of trans fatty acid increase LDL or bad cholesterol. They decrease HDL or good cholesterol, and they should be avoided in excess amount. Solubility of fatty acids, hydrocarbon chain. So there is no non-planar, poor water quality. They are longer chain, fewer double bonds, and the lower solubility. So short chain fatty acids, they are slightly soluble due to polar carboxyl group. Melting point of fatty acids depends on chain length and the degree of unsaturation. Saturated fatty acids are C12 to C24, so they are solid or waxy at 25 degrees Celsius. Unsaturated fatty acids, oily liquids at room temperature, and their lower melting point due to loose packing. Packing of fatty acid chains. Saturated fatty acids, they have straight chains and they are involved in tight packing. They have strong van der Waals forces. Unsaturated fatty acids, their chains are kinked due to cis double bonds and loosely packing. Weaker intermolecular forces are there. So fatty acids in blood, free fatty acids, they circulate bound to serum albumin, mostly present as derivative esters or amides. These derivatives lack charged bond COO group, that is carboxyl group, carboxylate group, and they are even less soluble in water. So this is the saturated fatty acids melting point of different saturated fatty acid, and these are the melting point of different unsaturated fatty acids. Triacylglycerols definition. Triacylglycerols are the simplest lipids formed from fatty acids. They are also called as triglycerides, fats, and neutral fats. Structure: they are composed of one glycerol molecule and three fatty acids. Fatty acids are attached to glycerol by ester linkage. Types of triacylglycerol. Simple triacylglycerols, all three fatty acids are identical. They're named after the fatty acid present. Example, uh, tristearine. And mixed triacylglycerols, they contain two or more different fatty acids, and mostly naturally occurring fats are mixed. For exact naming, the type and position of each fatty acid must be specified. So chemical nature, we know that there should be esterification. Esterification removes polar OH group of glycerol. This is glycerol. This is the OH groups. There are three OH groups, and polar carboxyl groups of fatty acids. There are three carboxyl groups in three different fatty acids. Hence, triacylglycerols are non-polar, hydrophobic, and they are insoluble in water. Density and phase separation. Lipids have lower specific gravity than water, therefore oil, we know, floats on water. Oil and water mixtures form two layers there. Storage of triacylglycerols in eukaryotic cells, we know it's stored as microscopic oil droplets in cytosol. They act as energy depot. In vertebrates, they stored in specialized cells called adipocytes or fat cells, and fat droplets almost fill the entire cell. In plants, it stores as oil in seeds. It provides energy during seed germination. So, what is mobilization of fat? We know that mobilization of fat, lipase enzymes hydrolyze triacylglycerols. The products are free fatty acids and glycerol. Their fatty acids are transported to tissues where they are used as fuel. Why triacylglycerols are ideal energy stores? Triacylglycerols are high energy yield. The fatty acids are more reduced than the carbohydrates, we know, and the oxidation of fats yield more than twice the energy per gram compared to carbohydrates. The second point is no water hydration. Triacylglycerols are hydrophobic and they are stored without water. So polysaccharides store 2 gram water per gram and increasing body weight. Fat storage in human. Fat tissue located under the skin, abdominal cavity, and mammary glands. A moderate obese person can store 15 to 20 kg fat that survive for months using fat reserves. Human male and human female, there are different types of fat deposition in their body. We can find that in case of human male, there is a apple shaped body fat distribution around the abdomen, and this apple shaped is the indicates the more visceral fat should be there, and there is a higher risk of weight related health problems. But in case of female, there is a pear shaped fat deposition in the body, and it present around the thigh and buttocks. Pear shaped, it has less visceral fat and lower risk of weight related health problems. So glycogen stores, we know that it lasts less than 1 day. Additional functions of triacylglycerols. First one is the thermal insulation. We know that fat under skin prevents heat loss. It is found in seals, walruses, penguins, polar mammals, etc. Hibernation, in animals like bears, fat provides energy plus insulation during hibernation period. Buoyancy. In sperm whales, triacylglycerol and waxes help in buoyancy control during deep dives. We know nature of dietary fats. We know that there are four types of dietary fats explained. Saturated fats, there is no double bond in the chemical structure. All our single bonds are present. Monounsaturated fatty acid, there is a single double bond structure. Polyunsaturated, more than one double bond. And trans fat, there must be double bonds, and the hydrogen atoms on both side of the present on both side of the double bond and both side of the chain. So we know that natural fats are mixture of simple and mixed triacylglycerols, and fatty acid chains vary in chain length and degree of saturation. So there are variation in the degree of saturation. Vegetable oils versus animal fats. We know that vegetable oils are rich in unsaturated fatty acids and they are liquid at room temperature. Examples: corn oil, coconut oil, olive oil, groundnut oil, etc. And animal fats, they are rich in saturated fatty acids. They are so they are solid at room temperature. An example: tristearin, that is beef fat. So, what is hydrogenation of oils? Industrial process that converts oils into solid fat. This is the hydrogenation reactions occurs in presence of nickel catalyst. This is the vegetable oil which is in unsaturated condition. They can be saturated vegetable ghee. So, they reduces double bonds and produces trans fatty acids. Rancidity of fat, they occur when fats are exposed to oxygen for long periods. They are caused by oxidative breakdown of unsaturated fatty acids. They produces aldehydes and short chain carboxylic acids. They results in bad smell and unpleasant taste. So rancidity of fats and oils causes a disagreeable smell, texture or appearance. So this is hydrolytic rancidity, and this reaction occurs more quickly in the presence of heat and moisture. It is catalyzed by the enzyme lipase. So the rancid smell due to the release of fatty acids. So hydrolytic rancidity can be reduced by refrigeration. Biological waxes are esters formed from long chain fatty acids, that is C14 to C36, and long chain alcohols C16 to C30. So fatty acids may be saturated or unsaturated. Physical properties: they are highly hydrophobic, they are water repellent, so firm and solid at room temperature. High melting points indicates that there should be usually between 60 to 100 degrees Celsius, and they are higher than triacyl glycerols. Role as energy storage: in plankton, microscopic marine organisms, their waxes are the main stored form of metabolic fuel, and plankton form the base of the marine food chain. Protective functions of waxes: due to their water repellent nature, waxes prevent water loss and it protect surface from damage. Waxes in animals. In vertebrates, they are secreted by skin glands. Functions: they protect skin and hair, keep skin soft, flexible and waterproof, and they act as a lubricant. So this is there are bee wax, waxes present in birds feather, waxes present in whales, spermaceti wax in found in sperm whale. So these are the animal wax. In birds, they are secreted by preen glands, especially important in water birds. They keep feathers waterproof, flexible, and well conditioned. Waxes in plants, they present as a thick waxy coating on leaves. We know they are seen in plants like holly, rhododendron, poison ivy, and many tropical plants like carnauba palm. They're basically found in the plant leaf surface. So we must know the functions of waxes in plants. It reduces excessive water loss or transpiration, and it protects against parasites and pathogens. Industrial and commercial uses of biological waxes: they are widely used in pharmaceutical industry to produce ointments, creams, medicinal formulations, cosmetic industry like lotions, lip balms, skincare products. Other applications include polishes, waterproofing agents. Examples of natural waxes are lanolin, which are obtained from sheep or lamb's wool; beeswax, they are produced by honeybees; carnauba wax, they are obtained from Brazilian palm tree; and spermaceti wax, they are obtained from spermaceti oil. So these are the benefits of beeswax. It acts as skin enhancer or skin moisturizer. They act as liver protector, cholesterol stabilizer, pain reliever, and anti-inflammatory. They have a leaf healer property, acne treatment, and the stretch mark eliminator. So now we will study about the glycerophospholipids. Glycerophospholipids are basically major membrane lipids. They built on a glycerol backbone. So there in chemical structure you can find there is a glycerol, two fatty acid chain, and one phosphate molecule will be attached to the glycerol, and another alcohol must be attached to the phosphate. So basic structure: we know that glycerol has three carbons, C1 and C2. That is C1 and C2 are attached to two fatty acid by ester bonds. These are the ester bonds. These are the ester bonds. And C3, that is the carbon 3, they are attached to the phosphate group. So a polar or charged head group is attached to a phosphate by a phosphodiester bond. Chirality of the glycerol. Free glycerol is prochiral, that is no asymmetric carbon. So attachment of phosphate makes it chiral. So its correct names are L-glycerol 3-phosphate or D-glycerol 1-phosphate or SN-glycerol 3-phosphate, which is commonly used. Naming of glycerophospholipids, they named as derivatives of phosphatidic acids. The name depends on the polar head group. Examples include phosphatidylcholine or PC found on the head group, that is choline, phosphatidylethanolamine or PE, the head group is ethanolamine, phosphatidylserine is PS, and phosphatidylinositol, that is PIP2. Charge on head groups at neutral pH, their phosphate group always negatively charged and their head group may be negative, so phosphatidylinositol 4,5-bisphosphate. It can be neutral or zwitterionic, that is phosphatidylserine, and it must be positive, that is phosphatidylcholine, phosphatidylethanolamine, etc. So this is the polarity of the phospholipids. We know the fatty acid composition. Fatty acids can vary. Many molecular species of the same phospholipid. The distribution depends on organism, tissue type, or cell membrane. And general pattern: C1 position indicates C16 or C18 saturated fatty acid, and C2 position should be there C18 to C20 unsaturated fatty acids. Ether lipids definition: they are modified glycerophospholipids. One fatty acid chain is attached by an ether bond instead of an ester bond. So types of ether lipids: alkyl ether lipids, they are saturated ether chain. Plasmalogen, the ether chain with a double bond between C1 and C2. It is abundant in vertebrate heart tissues. There is 50% plasmalogen. Halophilic bacteria, they are ciliated protists, some invertebrates, etc. So what are the significance of ether lipids? We must know that ether lipids are more resistant to hydrolysis. They are resistant to phospholipid action. So exact function of ether lipid is not fully known. So example, there is a this is a chemical structure of ether lipid, and examples of ether lipid include the PAF or platelet activating factor. It's a biologically active ether lipid. It is released from the basophil WBC, basophil white blood cell, and their functions include platelet aggregation, serotonin release, which acts as vasoconstrictor. It acts on liver, heart, smooth muscle, uterus, lungs, and they are important in inflammation and allergic reactions. Galactolipids, they're found in plant membrane. The definition: membrane lipids predominant in plants, that is galactolipid. One or two galactose galactose molecule attached to the C3 of 1,2-diacylglycerol by glycosidic linkage. So location: it is found in the thylakoid membrane of chloroplast. Importance: it constitutes 70 to 80% of total membrane lipids of the vascular plants and probably the most abundant membrane lipids in the biosphere. Archaeal membrane lipids, they're found in archaebacteria. Archaea live in extreme environments. They have high temperature, low pH, and high salt concentration. Structure: so lipids contain long chain, approximately 32 carbon branched hydrocarbons. The chains are linked to glycerol by ether bonds. Significance: ether bonds are more stable at high temperature. They are resistant to acidic conditions, unlike eubacteria and eukaryotes, which are ester linked lipids. Sphingolipids, fourth major class of membrane lipids. Key features: they have one polar head group and two nonpolar tails. They do not contain glycerol, and structurally they are different from glycerophospholipids or galactolipids. So the basic components of sphingolipids, they are made up of sphingosine, which is a long chain amino alcohol, and it has four sphingine molecule. So one long chain fatty acid and a polar head group. So this is the sphingosine. There is a fatty acid. There is a phosphate group, and there is a choline unit, a polar head group, and head group may be attached to glycosidic bond or glycolipid, and phosphodiester bond or phospholipids. Sphingosine backbone: sphingosine has C1, C2, and C3. These carbons are structurally analogous to glycerol carbon. They are fatty acid attached to amino group at C2 via the amide linkage. So this is the sphingosine and this is the fatty acid. These are basic components of the sphingolipids. Ceramide, which is the parent molecule. When a fatty acid attaches to sphingosine, it is ceramide. This is the sphingosine and the fatty acid residue with variable length. And ceramide is basically structurally similar to diacylglycerol, we know, and the structural parent of all sphingolipids. So this is the parent molecule of the all sphingolipids. So subclasses of sphingolipids, all sphingolipids are ceramide derivative differing in their head groups. Three main subclasses: sphingomyelins, neutral sphingolipids, and gangliosides. Sphingomyelins, their head group, there should be phosphatidyl phosphocholine or phosphoethanolamine. They're classified as phospholipids. Properties: they're similar to phosphatidylcholine, so there is no net charge on head group. Their location: plasma membrane on animal cells, and myelin sheath are present on neurons. Electrical insulation of nerve fibers. Glycosphingolipids, they are located mainly on outer surface of plasma membrane. Their head group on one or more sugars. Sugars are attached directly to bond OH at C1 of ceramide, and there is no phosphate group. Cerebrosides, that is simplest glycosphingolipids, they contain simple sugar. Their type includes galactocerebrosides, which are neutral tissue, and glucocerebrosides, they are non-neural tissues. Neutral at pH 7. So globosides, they are neutral glycosphingolipids. They contain two or more sugar. The common sugars include D-glucose, D-galactose, and N-acetyl D-galactosamine. So there is no net charge at pH 7. So neutral glycolipids include cerebrosides or globosides. They are called neutral glycolipids, and reason there is no net charge at physiological pH. So that is the structure of cerebrosides. That is the simplest glycosphingolipids, and this is globoside. This is a neutral glycosphingolipids. So now we should study about the gangliosides. There are most complex sphingolipids, that is the structure of gangliosides. Their head groups include oligosaccharides, one or more sialic acid residues. Sialic acid provides negative charge at pH 7. So classification of gangliosides, they are based on number of sialic acid residue. GM, there is one sialic acid, ganglioside monocyte, and GT, ganglioside di, there is two sialic acid, GD for di, two sialic acid, GT, three sialic acid tri, and Q, there is four sialic acid or quad. Sterols in different organisms. We know that in animals the sterols are cholesterol. In plants they are stigmasterol. In fungi they are ergosterol. In bacteria basically cannot synthesize sterols, and some can incorporate sterols from the environment. So this is the basic structure of steroids, which are characterized by a four-ring structure. They are found in cell membrane, where they influence the membrane's fluidity, and they can also act as a hormone in the body. So biosynthesis of sterols. Sterols of all eukaryotes are synthesized from 5-carbon isoprene units, and this pathway is common to all eukaryotic sterols. Functions of sterols in membrane. We know that they regulate membrane fluidity, membrane stability in the cell membrane structure. They prevent membrane from becoming too rigid at low temperature and too fluid at high temperature. So sterols as precursors of important molecules, they are not just membrane components, they also give rise to biologically active molecules. So they can produce steroid hormones. Steroid hormones are derived from cholesterol. Examples include sex hormone, adrenal hormone, etc. Their functions are they act as a powerful biological signal, and they regulate gene expression. Bile acids are also polar derivative of cholesterol. Their functions include they act as detergents in the intestine, they emulsify dietary fats, and they make fats which are accessible to digestive lipids. So, my dear students, we have now seen how lipids, though often called simplest fat, are vital architects of life, from forming the cell membrane with phospholipids to storing energy as fatty acids and regulating body functions through steroids. Lipids play key structural, energetic, and signaling roles. As you prepare for university and competitive exams like CSIR-NET, remember that understanding their structure and biological significance is fundamental to mastering biochemistry. So keep revising, stay curious, and continue exploring the molecular wonders of life. Thank you, and see you in the next class. Bye.